And we have today also one more participant. It's Anastasia Stepanyshyn, student, Franko National University of Lviv. So Anastasia, you can start your screen.
1 minute. I hope you're seeing my presentation. Yes, everything is working correctly. Super. So good morning, everyone. My name is Anastasia Stepanyshyn, and the title of my presentation is Developing Genetic Tools for Manipulating Streptomyces ROSEOCHROMOGENES, NRRL 3504, Genome for Increasing CLOROBIOCIN Production. I will start off with some general information about Streptomyces, and as you may know, Streptomyces is a genus of gram-positive bacteria, and there's the soil-dwelling microorganisms that possess mycelia growth. Their genome is characterized by high GC content, but for us humans, the most important detail about them is the ability of Streptomycetes to produce a wide variety of antibiotics such as kanamycin, chloramphenicol, and apromycin that you can see on this slide. And speaking of antibiotics, next I will talk about Streptomyces ROSEOCHROMOGENES. This strain is a known producer of aminocoumarin antibiotic CLOROBIOCIN, and this antibiotic consists of 3 rings and is halogenated, as you can also see on the slide. And it is effective against multi-resistant gram-negative bacteria when combined with other antibiotics. Previously in literature, it was mentioned that this strain is difficult to manipulate. Therefore, in the past, for genetic manipulation, heterologous expression was used. But our goal was to develop new approaches for genetic manipulation of the producer strain itself, and further increasing the CLOROBIOCIN production by this strain. To achieve this, first of all, we tested the conditions for optimal plasmid transfer into this strain. For that, we used 4 different vectors. 3 of them are based on phages, that is P-Saga, it is FEC31 based. P-Raga1, this is FBT1 based plasmid. P-Kaga1, this is based on an integrative vector PSG5. And the last one, P-Saga2, it is VWB based, this is also a phage based plasmid. All of this plasmid carried gene GUS-A under the control of constitutive promoter, and GUS-A is responsible for beta-glucuronidase activity in E. coli. So because of that, we were able to verify the successful integration of plasmids into our transconjugates genome via performing glucuronidase assay. Because the transconjugates, they were able to transform X-gluc when applied to the surface to 5,5-D-promo for 4-dichloroindigo. And also we verified them via PCR. Moreover, we also tested the conditions that are optimal for the conjugation. For that, we tested 2 media. One is soy flour mannitol media, and other is oatmeal media or ESP3 in the presence of magnesium chloride or without. And we have determined that the highest transfer rate was observed when using the FEC31 base plasmid. And for optimal conjugation conditions, the best results were achieved on oatmeal medium without the presence of magnesium chloride. Our next step was constructing a collection of plasmids, 12 in total. 2 of them were carrying 2 genes from Streptomyces albidoflavus. This is ADPA and VLDA. And as we knew, those genes in Streptomyces albidoflavus genome show high similarity to their counterparts in Streptomyces ROSEOCHROMOGENES genome. And why this set of genes? Because first one codes, so ADPA, it codes for global regulator of secondary biosynthesis in Streptomyces, and overall has a positive impact on the biosynthesis of antibiotics and other substances that are produced by Streptomycetes. And the latter is a gene for lysine tRNA, which is tightly intertwined with ADPA as well. And next 10 plasmids can be split into 2 groups, as you can see on the slide. First group consists of expression vectors carrying additional copy of CLOG gene. This gene is coding for path-specific regulator of CLOROBIOCIN biosynthesis pathway. And second group consists of different expression platforms carrying NOVG, and NOVG is an ortholog of CLOG that is responsible for regulation of novobiocin biosynthesis in Streptomyces niveus. After we constructed these plasmids and successfully transferred them into our production strain of Streptomyces ROSEOCHROMOGENES wild type, we had to estimate chlorobiocin production in transconjugates. For that, transconjugates were cultivated in GEM liquid medium for 120 hours, and each 24 hours a sample of cultural broth was taken from each flask. And in the end, we extracted the antibiotics from the samples and dissolved them in ethyl acetate. And to test chlorobiocin activity, we performed disk diffusion assay against a gram-negative test strain that was Micrococcus luteus. And the results show that overexpression of both VLDA and ADPA had positive impact on chlorobiocin production compared to wild-type strain, as you can see on the slide. And overexpression of both regulatory genes CLOG and NOG also had a really positive impact on levels of biosynthesis, of production of chlorobiocin. And the best results were observed while overexpressing NOVG under control of QMAT-inducible promoter in presence of 9 milligrams per milliliter of QMAT. This is the plate I on the slide. And to summarize it up, we were able to achieve increase of production of chlorobiocin in transconjugates to up to 32-fold. As demonstrated by NRRL 3504 carrying a plasmid PNOG 103 in presence of QMAT. And conclusions of this work is that we were able to develop genetic tools for manipulating chlorobiocin producer Streptomyces ROSEOCHROMOGENES and successfully transferred 12 plasmids carrying additional copies of CLOG and NOG, as well as VLDA and ADPA. And the highest chlorobiocin production was demonstrated by recombinant S ROSEOCHROMOGENES strain carrying additional copy of NOVG under control of QMAT-inducible promoter P21 in presence of 9 milligram per milliliter QMAT. So thank you for your patience. Maybe you have some questions about my
Thank you so much, Anastasia. So we have a question from Julia, yours first one.
Thank you. It was an amazing talk, really. I honestly have only technical questions. My first question is, why do you call them transconjugates? What is the process of implementing this DNA into the cell? Isn't it the transformation?
No, because we're working with Streptomyces and they're really difficult to transformate, the host cells. So we are using the intergenic conjugation. So this is between Streptomyces and E. coli.
Aha, okay. But you use phage-based vectors, but they are already like transformed into E. coli, and then you have this conjugation between E. coli and Streptomyces.
Yes.
Okay, and second, also only technical question.
Okay, sure.
When you work with Streptomyces, your plasmids, do they contain the same promoters as for E. coli vectors?
Well, they're from the beginning, they're built for expressing in Streptomyces cells. So I don't think that the promoters are the same as for E. coli, because most of our plasmids that were shown here, they contain their own native promoters from the, for example, genome of Streptomyces albidoflavus.
Okay, so generally you have different one, but did
Yeah.
you compare how much they differ from E. coli?
Actually, I don't think so that we did that, but this is a really nice insight. So thank you for an idea.
Okay, thank you. Very good talk. Thank you.
Thank you so much, Julia. So Anastasia, I also have a question. Did this all manipulation, this one construct geneticals was conducted everything, this one methodology in your university in Lviv, or something was conducted abroad?
I think only one thing was conducted abroad. It was the LCMS measuring of CLOROBIOCIN production. So as you can see here, this is where we got our information about the fold change. So we do not have the capability to do that in our university. So we had to ask our partners for that part.
Thank you so much. So you also can, any questions more? Guys, expert committee. So Anastasia, thank you so much. You can start to share the screen. So